Welcome everyone, thank you for coming. It is my great pleasure to introduce our today's speaker, who's Professor John Marko from Northwestern University. John has completed his PhD at MIT, followed by postdoctoral training here at New York City at Rockefeller University, where he worked with Albert Liebschaber and Eric Sigia. And some of you might be familiar with his, by now, iconic experiments of micropipette aspirations of mitotic chromosomes, where he was, for the first time, for the first person ever, pulling on the real chromosomes and looking at their elastic and mechanic response. He has a large interdisciplinary group in Chicago at he disagrees, at Northwestern.
I've been trying to make it smaller.
Oh, I see. University, where he studies all things exciting and related to DNA and mostly chromosomes. His big focus used to be mitosis and meiosis, so the chromosomes condensed during cell division, but since a few years ago he started also to work on the interphase nuclei, so when your genome is actually decondensed. And also he has a huge body of work on naked DNA and on the physical properties of the DNA as an interesting polyelectrolyte, so charged polymer. So without further ado, I can't wait to hear the newest and the latest. John.
All right, thank you. Well, and thank you for the invitation to NYU. It's great to visit you guys again. And okay, so this is obviously an advertisement for a Gordon conference on chromosome dynamics. The deadline to apply is about a month away, or a few weeks away, and the conference is in June in beautiful Maine. So we're at a moment in biology history where people worried about genetics and molecular organization of chromosomes are also very interested in their physical organization. I mean physical with meaning physics. This is a physics audience, and a lot of the people at this meeting are going to be talking about things very relevant to problems in physics, which is what I'm going to more or less talk about. So increasingly in biology, biologists like to think about mechanistic accounts of cellular machinery, molecular machinery being the lowest level of how we would think about the operation of the cell. And a physics audience knows, at least for simple machines, we need to understand three different things. Two of them, biologists, you're never going to compete with them. They're very good at measuring things in space and time using very advanced imaging and mapping techniques. Now it's quite possible to map where sort of all of the molecules are in the cell at near to nanometer kinds of precision in one way or the other. But we should remember, and physicists always can remember, that maps and kinematics are really not enough to understand how a machine works. You need to understand forces as well. So I think this is one of the principal ways that physicists can make a big impact in biology is by introducing biologists to the idea that we have to understand the forces that are being applied in, for example, a cell division during mitosis, and what the origin of those forces are. In this case, microtubules are attaching to the chromosomes, and motors along those microtubules are eventually pulling the two replicated chromatids in each mitotic chromosome to the two sides of the cell. In this, just a point, this beautiful movie was made by one of my first graduate student, Michael Poyer, and it's from cells which have very large nuclei, about 50 microns across here. You can actually see the nuclei without a microscope when these cells are growing in culture. And each one of those chromosomes you saw being pulled across the cell actually has inside it a DNA molecule that's about the size of the entire human genome, and these are the lowly nukes. So humans don't have any kind of stranglehold on length of DNA. So what I'm going to be telling you about today is chromosome organization from a biophysical point of view. Basically, a chromosome, we talk about a long DNA molecule. It, of course, has coding information for genes along it in its base pair sequence. And even in the simplest living organisms, bacteria, single-celled organisms, we have chromosomes which are on the millimeter range of length, millions of base pairs long. And I've just shown you a cell division in an organism that has meter-long DNA molecules. And of course, just shoving all that DNA inside the cell is not going to be very useful. It's brought to life as a chromosome by many proteins bound along the length of that chromosome. I'm going to be telling you a little bit about the action of a few species of those proteins in helping to spatially and temporally organize the chromosome. So chromosomes are highly dynamic. They're well organized, though, in a regime where there's a lot of stochasticity and a lot of randomness, as you've probably heard from Alexandra and others around NYU. There's a lot of dynamic dynamic nature to chromosomes, but yet they stay well organized geometrically and also have to be separated from one another. They're organized topologically. So I'm going to be telling you a little bit about chromosomes at the metaphase of, you can think of animal cell division, where the chromosomes are compacted along their length into long noodle-like structures which allow them to be manipulated inside the cell and separated from one another. And I think there's a few things I want to emphasize, that this is accomplished by some kind of magic crosslinkers, which I'll tell you a little bit about, but not in the usual way that we think about crosslinkers acting to stick polymers together into a condensed glob. There's no condensation in the usual sense in this type of compaction, and there is not a skeleton of protein in the way that many biology textbooks portray for these types of chromosomes. Then I'm going to talk about nuclei. As Alexandra mentioned, we work on mechanics of nuclei, and tell you a little bit about what we can infer about the different contributions of the boundary of the nucleus and the interior, the DNA and the DNA complex with proteins that we call chromatin in the interior of the nucleus. And in fact, the stiffness of the nucleus and its shape are tuned by properties of the chromatin in the interior of the nucleus. And this is in fact connected with various types of pathological disease-like states. So I've already told you that we have long strings in a bacterial cell, millimeter lengths of DNA, and a newt cell, 20 meters of DNA, roughly speaking. And so this game is a kind of a game of understanding how very long flexible strings can be packed into small boxes, and where we have thermal fluctuations and non-thermal active fluctuations generated by the active cell machinery, which, remember, inside a living cell is a highly non-equilibrium environment where there's plenty of energy available in the form of things like ATP, which are highly energetic molecules which can be used to power nanometer-size enzymes. It's a very nice area for studies of people in condensed matter physics, particularly towards the statistical mechanical end of condensed matter physics. You know that human cells and other cells with nuclei go through a cell cycle where you pass from a stage of the cell where the chromosomes are, as Alexandra mentioned, kind of dispersed in the interior of the cell. There's a couple of nucleoli inside the nucleus where ribosomes are being made. But the chromatin in the interphase is spread out inside the nucleus and is actively being read to make eventually messenger RNAs that are exported to ribosomes to make proteins. But then something magic happens. The chromosomes start to appear as long, flexible strings that gradually get shorter and thicker and stiffer, and we reach the so-called metaphase, where the boundary of the nucleus disappears and we have chromosomes floating around in the interior of the cell. As you saw in the movie, the chromosomes get split apart. They're actually pairs of side-by-side replicated chromosomes at this point to the two sides of the cell, and then form the two nuclei of the daughter pair of cells. And then this process is how cells grow in our bodies and in all eukaryotic living organisms. Okay, so we do experiments isolating objects from cells, and I'm going to tell you about chromosomes, metaphase chromosomes, and also nuclei. And basically the approach we use is to work on a microscope. So there's a dish, a shallow dish here where cells are growing, and we can open a cell and grab a chromosome with a long, thin glass pipette, which tapers down to micron dimensions, which we can control with motorized manipulators. And by choosing the taper of this pipette, we can also use these objects, these glass pipettes, as force sensors. So when we move one pipette, if something like a chromosome is attached between that pipette and another pipette, we can move one pipette and watch the other pipette bend, and we can calibrate that bending stiffness to do force measurement. And here's the mechanical response of a metaphase chromosome, which started out being 16 microns long. This is a rather large newt chromosome being extended slowly, and you see the force go up in piconewtons on the sort of roughly nanonewton scale. And here we're increasing the length of the chromosome by about a factor of 50%, and then we can bring it back down. And you see it's nearly linear elasticity and reversible elasticity over this very large strain range. This is already a sign that what we're working with is a kind of polymer gel with very well-defined elastic response. All right, so to give you an idea of how this type of experiment actually works, my student Mike Poirier made a beautiful movie of isolation of the genome of a newt cell, which is now at the metaphase, so that you see the individual rods of chromosome floating around inside the cell, and he's just puffing a little bit of a surfactant. It's a dilute solution of a surfactant that will just destabilize this region of the cell membrane, and you'll see a whole genome come out of the cell. At this stage of the cell cycle, the interior of the cell, cytoplasm, is rather liquid, and there's very little impediment to just releasing the genome from the cell into the surrounding liquid where the cells are growing. And so there comes the genome out of the cell, and you see it's like a bunch of soft noodles. And I want you to realize a few things from this picture. One, those soft noodles are not sticking to each other particularly strongly. They're individualized and separated units. They're flexible, but they're flexible on a scale. The scale bar here is that this is tens of maybe 30 microns across the whole genome here. And you can see that the chromosomes are starting to spread away from one another, but they're retaining their individual string-like shape. And each one of these chromosomes you can kind of pick up is actually a pair of side-by-side chromatids, which are the replicated pairs of chromosomes attached together. It's just the name for them. The other thing that you immediately see when you do this type of experiment is that the chromosomes are attached to each other. And this is something that's very poorly understood, but if we see in every type of genome that we isolate, there are thin fibers of chromatin linking the different replicated chromosomes. These are not the connections between replicated chromatids that you see at the end of cell division. These are connections between different non-homologous chromosomes, the non-identical chromosomes of the genome, and they're sort of not supposed to be there according to classical physiology and cell biology. And in fact, Mike is going to grab, with a little bit of osmotic pressure, the end of this chromosome, and you'll see that it will stick to the glass pipette very well, but the chromosomes don't really stick to each other. So they're not condensed in the usual sense of there being a strong surface tension holding chromosomes together. And you'll see as he pulls this chromosome away from the rest of the genome that there's an invisible, hard to see, filament. We know these filaments are about 100 nanometers thick, linking the chromosomes together. In fact, they're very strong. This whole chromosome will start to stretch as we extend this invisible filament. For us, this is just an annoyance for this particular experiment. So we're going to cut this filament by moving another pipette close, and I'm just going to zoom to sort of the later stage of the movie where a second pipette is attached. And the pair of pipettes is moved away from the genome. You can see this pipette's bouncing around. It's a very floppy force measuring pipette, and then that filament breaks, and we can do an individual chromosome experiment. So what do we know? I've already shown you data showing that the spring constant of a chromosome, its force constant as we say it, the force needed to double the length of a chromosome for a thick newt chromosome, about 2 microns thick, and this one is about 20 microns long. The force being exerted on this force measuring pipette for doubling the length of the chromosome, this chromosome, is around a nanonewton, okay, about 1,000 piconewtons. For a sense of scale, a piconewton is about a kBt per nanometer, is 4 piconewtons. That's the scale of individual biomolecule interactions inside the cell, something that can overcome thermal fluctuations and is of nanometer dimensions. While a nanonewton is 1,000 piconewtons, it would be naturally the force scale associated with stretching thousands of biomolecules at once, which is what we're doing in this experiment. So, okay, for some years we did experiments on the chromosomes from newts, and then we started to work on the harder problem of doing experiments on human chromosomes, which there's a newt chromosome, there's the length scale bar, here's about the same length scale bar. You see the human chromosomes are thinner and shorter, harder to work with. Nevertheless, they show the same linear out and back force response, but on a much lower force scale. The same kind of material, just thinner piece of it, so you have a lower force constant. In fact, both the newts and the humans and every other chromosome from eukaryotes we've studied has a Young's modulus, if you like, the spring constant per volume of about 400 pascals. So if you want to think about chromosomes as soft matter, they're elastic solids, same as a piece of steel. It's just that their Young's modulus isn't a gigapascal or 10 gigapascals, it's a few hundred pascals. Okay? There's also a very large internal viscosity of the metaphase chromosome. We've done measurements of this some years ago, and it's on the order of 100 pascal seconds. You'll remember the water is about 10 to the minus 3 pascal seconds. So in fact, in these experiments, we have to, the water viscosity plays no role in the viscous damping. It's actually the rearrangement of the fibers inside the chromosome that plays a key role. All right, good. So this is my world where we work thinking about chromosome organization. We have DNA. Some of you know that DNA, the first level of folding in human cells or eukaryote cells, is due to the wrapping of DNA around clusters of proteins called nucleosome, called histone proteins. The structure is called a nucleosome. This is a basic repeat unit of about 10 nanometers thickness. All the stuff in this picture is roughly to scale. There's the 2 nanometer thick DNA, 10 nanometer thick nucleosomes, and they can form fibers which are somewhat thicker. Then the DNA inside here can interact with a whole variety of proteins, and I've shown a few proteins that are sort of relevant to organization of chromosomes, but I want to bring your attention to this protein right here, topoisomerase 2. So when you have long strings packed into small boxes, you can easily get tangles and topological impediments to motion. Topo2 is very important in overcoming this. In fact, drugs against Topo2 are potent, are some of our most potent biocidal drugs. Some of you have heard of ciprofloxacin. That's an antibiotic that targets the version of Topo2 in bacteria. Other poisons against Topo2 are commonly used as chemotherapeutics as well, as cancer chemotherapeutics. So what this thing can do is cut one piece of DNA and pass another piece of double-stranded DNA through the resulting gap and then seal up the original DNA. That is the key move that you need to make. Any knot theorist knows that will allow you to tie or untie any knot. Okay, if you can make that type 3 Redemeisters move, and it's kind of cute that Topo2 does exactly what the knot theorists need to change the topology of their knots. But Topo2 will just as happily make knots as unmake knots, and it needs to be told what to do by some other enzyme. And what us and many other people in the field, I think is that these proteins, which are common to all living things, unlike nucleosomes, which are only in eukaryotes, these types of protein complexes, which are very large enough to pass DNA fibers through, we think, the interior loops of them, and I'll be focusing on this thing condensin today, are made of tripartite rings, hydrolyze ATP. Topo2 also hydrolyzes ATP in its reaction cycle. Condensin hydrolyzes ATP in some way to somehow guide Topo2 to remove the entanglements from different chromosomes and to fold up chromosomes during mitosis and to do other jobs where DNA needs to be organized into loops. So I'll be talking a little bit about this experiment. Now, many biology books will tell you that there's a kind of ribbon of protein along the interior of the chromosome that organizes the metaphase chromosome. So Mike did in his PhD thesis some time ago, in a different kind of, not quite a different millennium, did an experiment that's kind of an experiment only a physicist could do because it's so simple. He took a metaphase chromosome and then he exposed it to enzymes that cut DNA with no other perturbation. And lo and behold, what happens is the chromosome gets cut through, indicating that there's no internal skeleton of non-DNA that can withstand this cutting process. I'm going to run that again. And the chromosome loses its elastic stiffness. There's actually a little bit of stiffness, a little bit of tension on the chromosome. At the beginning of this movie, you'll see the force measuring pipette, which is, I think, this one, relax as the cutting proceeds. And the whole chromosome changes from being a rod, it's no longer a rod, to a thing that looks like a droplet, and this droplet will eventually evaporate. It again shows us there's no real condensation, no sticking of the chromatin to itself in the chromosome, and all of the elastic stiffness, the mechanics, the shape of the chromosome depends on DNA and not on protein, except in connection through DNA. So we conclude, as a soft matter person would, that the structure in the interior of the chromosome must be at least topologically like this, with linker proteins of some kind, presumably proteins, not DNA, which are linking the contiguous DNA fibers together so that when you cut the DNA up, the whole thing falls apart. Very simple topological argument. All right, good. Okay, and human chromosomes actually completely disappear when you expose them. These are to enzymes, I won't go into the details, that don't cut every DNA site. They cut a restricted set of either 4 base targets or 6 base targets for taking the chromosome apart. Well, so there's plenty of evidence that these crosslinkers are the condensin SMC complexes that I talked about earlier. And maybe I'll just draw a picture of condensin. It kind of looks like this. This is called SMC2, this protein. This is called SMC4, and this thing here is called CAT-H. And then there are a couple of additional proteins. I want you to just remember that there's this tripartite ring, and the ATP binding and hydrolysis, the motor part of them, is on these ends right there. All right, good. So my student, Mina-san, did a series of quite nice experiments combining biophysical and genetic techniques to look at the role of condensin in organizing metaphase chromosomes. And this was actually fairly recently published. And what she did, first of all, is we have tools in molecular biology where we can decorate specific proteins using antibodies. So we can take a chromosome out of the cell and then expose it to antibodies that bind, for example, SMC2. And in fact, you're looking at images of SMC2 along a metaphase chromosome that isn't stretched out. And you see that there's a kind of string of stuff along the length of the chromosome. You might conclude that that is a kind of scaffold to which the DNA is attached. But if you stretch out the chromosome, you see that there's a strong modulation of SMC2 rich regions, we sometimes call condensation centers, separated by regions with apparently almost no SMC2 at all. So we can directly see this broken up structure of the supposed protein scaffold into individual clusters of SMC2 protein. And a number of other labs are coming to the same conclusion about this, that the SMC2 condensin complexes are not connected together. And in fact, we've done recent experiments using fancy cells that actually carry intrinsic fluorescent tags along every SMC2, and we can see similar kinds of patterns along mildly stretched chromosomes, which are indicative of a inhomogeneous crosslinker distribution along the metaphase chromosome. All right, so a common genetic trick that can be used is to trick the cell into destroying the messenger RNAs for a specific protein by infecting it with RNAs that are basically complementary to those messenger RNAs. And the cell thinks then that its own messenger RNAs are a viral infection, and it wipes them out. So the result is, after some time, you can't make any proteins after the messenger RNAs are destroyed. After some time, the protein level of that particular protein goes down, and that's called interfering RNA interference, and we're using so-called small interfering RNA molecules. So there's a normal cell growing in culture, we call it wild type, untreated. If we treat with these RNA molecules that cause the cell to self-destruct its messenger RNAs, after some time, it takes about a couple of cell divisions to get rid of all of that protein, you'll see that there's no SMC2 left. You can see that actually there are two types of condensin. There's some condensin in the nucleus, and then there's condensin in the exterior of the nucleus. There are two types of condensin in most metazoan eukaryote cells, including human cells. All right, so now we can do the experiment of harvesting genomes after different amounts of time after Si Rna infection. So this is kind of nice. You get a string of results at different levels of depletion. So wild-type genome, you see individualized chromosomes pulled out of the cell. After 24 hours, those chromosomes start to be kind of misshapen and poorly organized. And after 48 hours, they're rather even more poorly organized. And after a long period where there's essentially no condensin, none of this SMC2 left, and these complexes are non-functional, what you have is all of the chromatin from the cell is organized into one big tangled mass. We've done nothing with topo2, nothing with any other enzyme. We've only killed this one component of the condensin complex. Very dramatic result. So these kinds of visualization results have been seen in a number of experiments since the late 90s when these proteins were first characterized. But I think they really emphasize that depletion of this protein leads to a failure in segregation of the chromosomes from one another, and also in the compaction, I shouldn't use the term condensation here, of the chromosomes into well-defined chromosome bodies. Condensin is a crosslinker, but it's also a segregation driving machine. Okay, so I want to talk a little bit more about that. So, okay, wild-type chromosomes, you take them out of the cell, actually we can play with them for hours. This is a 2-hour time course where untreated cell genome, it more or less looks the same after 2 hours. The SMC2 depleted chromosomes sort of can fold, possibly because of remnant SMC2 or possibly because of other protein complexes, into something that looks like a reasonable genome, but then that just falls apart after 2 hours. So these depleted chromosomes, depleted of this SMC2 protein, are not even stably folded at zero force. And this also shows you again in another experiment that the chromatin itself is not under what we would call in the soft matter field poor solvent conditions where it's sticking to itself. It is not condensed in the usual sense. So we like to use the word compaction instead for this type of organization. So just like any soft matter physics experiment, we can take the wild type cells, we can take the crosslinkers away, the modulus goes down, it's the Young's modulus, and the amount of fluorescence goes down from labeling the SMC2, so there's the depleted chromosomes, and here's the modulus, so we can cut it by about a factor of 10, and the mechanics of the chromosome is very, very sluggish at this point. But we can also trick the cell into loading way more condensin than it normally would during a cell division by killing the microtubules. It turns out if you use drugs that wipe out the microtubules that would normally pull the chromosome, be involved in pulling the chromosomes to the two sides of the cell sits there and it just keeps loading more and more condensin onto the chromosomes. They get more and more condensed, they get brighter and brighter in the antibody treatment, and the modulus goes up. So this line here actually has a slope of 1, which would be the sort of mean field dependence of the Young's modulus on the density of crosslinkers. So in some sense, this really behaves like a polymer gel in that the crosslinker goes up, the gel gets stiffer and tighter, and the modulus goes up. This is a static gel modulus. All right, so what's going on? How does condensin, do we think, fold up chromosomes? Well, one way to think about what's going on with chromatin in the nucleus, let's say that last experiment, we take away all of the condensin and we get very, very entangled chromatin. We could say, well, how does the entanglement level depend on how much polymer is in the nucleus? So we can imagine, a sphere bounding a bunch of polymers. Here's two polymers, a green polymer and a red polymer of some good length, confined in the interior of this artificial nucleus. And we can ask, what's the linking number? Just the simple Gaussian linking number, Gauss linking number of those two polymers. If you want to make that very mathematically solid, you can make the polymers into circles, but actually you can define a linking number perfectly well for open chains, because the ends tend to be near each other. It's not a big perturbation. It's not a big perturbation on the total number of links between the polymers to close them. Let's think about circles. Linking number 0, linking number 1, linking number -1. Linking number can be signed positively and negatively, as you guys remember, based on the orientation of the rings. So we're going to get positive linking number, we're going to get negative linking number. The thing we're interested in is not the total linking number, but in the spread of the linking number. If the polymers are all disentangled from each other, the linking number will be 0, and the average linking number squared will be 0. If they're highly entangled together, the linking number will be some large value, sum of positive and negative values, and it will random walk in, it will undergo a kind of random walk distribution, and the width of the linking number distribution will be large. So this is a good type of measure for how entangled polymers are together. So from basic polymer theory, and also from actual calculations, which one can do, just stuffing polymers into boxes, as published some years ago, you can show that first of all, by a scaling argument, the catenation number, as it's called, the linking number of separate DNA molecules in biology is called the catenation number rather than the linking number, which is of the two strands in the double helix. All right, so that linking number is basically for random polymer configuration inside the nucleus. You can imagine that linking number could be changed by units of plus or minus 1 by changing, actually by plus or minus 2, by changing a +1 link, contribution to the Gaussian linking number, that would be this crossing, to just switching these guys around like this. Doesn't perturb the rest of the polymer, and that would suggest to you, since you're always near another polymer segment, that the linking number squared would actually undergo a random walk in the length of the polymer. Capital N here is the length of these individual polymers. Little n here is just taking into account that if you have little n polymers, little n chromosomes in the nucleus, well, the linking number between any pair of polymers will just go down by that factor. And it turns out that this scaling theory is borne out by simulations, very simple mean field scaling theory. There's some dependence on the concentration of the polymers, which I won't go into. The key thing is that the catenation number squared goes up with polymer length, and it goes down with the number of polymers that are mixed together from looking at the linking of any pair of polymers. So this immediately tells you that there are two strategies to disentangle chromosomes. One is to make the polymers short, and maybe by cutting them up into a lot of polymers. So if you make a lot of short polymers, they're not going to be entangled together. And in fact, budding yeast appears to sort of follow this approach. It has a small genome cut into a lot of pieces, and each one of them is rather small. And it turns out that most of the yeast genome appears to be not entangled with itself. Well, however, human chromosomes are long, so what we have to do is figure out a way to affect the length of the polymer fibers to bring them down so that we'll have a disentangled genome. And there's a very simple way to do that. What we can do is we can gather up the polymers along their length. And so we call this process lengthwise compaction. It's not condensation, because this would be some kind of folding mechanism that would bring the polymer close to itself, nearby to itself, and make the statistical segments longer and thicker, thus reducing the effective length of our polymer. So this would bring capital N down by gathering the polymer up along its length, lengthwise compaction, but avoiding contacts and sticking together of polymers by interactions directly between different polymers. So if you read Degen's textbook on polymer physics, you'll learn about condensation of polymers driven by interactions, adhesion interactions between any pair of monomers. And here we're saying, no, we're not going to allow that. We're only going to allow interactions between nearby monomers along the same polymer. Okay, and that will accomplish compacting the polymers and disentangling them naturally. You'll go below the disentanglement level for the particular type of polymer, and they'll be disentangled from each other. So topo 2 will just remove you'll be driving the entanglements out of the polymers by compacting the chromosomes along their length. You have to do work to do this, and these things are actually ATPases, arguably somehow accomplished in this scheme. And it's still a bit obscure how this happens, but I'll be showing you in a minute how we think this is happening. So we have to always avoid sticking polymers together. Once we stick the polymers together willy-nilly, poor solvent conditions, we're just going to get highly entangled polymers, as is well known in polymer physics. Okay, so, all right, so how do we imagine these enzymes actually lengthwise compact chromosomes? This is a picture from a concert I was in Paris a few weeks ago, and some avant-garde composers who actually picked up on a proposal for how chromosomes are actually folded so as to compact them along their length. All right, so what we imagine is going on, based on ideas which are actually quite old in the molecular biology literature, is that if you had a machine that when it binds to DNA does the following. It binds locally to the same DNA polymer by binding to adjacent segments. It then uses ATP as a motor, has a motor function which uses ATP as the fuel to push a loop out of that region of where the motor has landed. And as this process takes place, you get a loop, and we call this extrusion of a loop. Loop extrusion is directional and uses ATP to drive a loop growing in size. The machine can remember what direction to push because it binds on DNA and captures this initial loop, and then it doesn't turn around. Its motors are aligned. All right, so but this is not enough. Turns out if you just have loop extrusion going, you'll get a string of sort of randomly sized loops and not very good polymer compaction. But if you allow dissociation, the spontaneous unbinding of these proteins after some time, stochastically, from the DNA that they're on, what will happen is that protein will come back and it will tend to land in the exterior region of a loop where there's a lot of non-loop DNA, and it will then extrude down to the bottom, reinforcing that loop. And so you have a kind of coarsening of loops where the small loops go away and the big loops get bigger and become anchored by multiple loop extruding enzymes. I'm going to show you how this works in just a minute. Yes, Dave.
I'm puzzling over something you just
said.
You said if you just have them forming these loops, it doesn't do what you need it to do. Is it obvious why not?
So imagine you just bind at time 0, right? A bunch of guys, and they're never going to dissociate.
Yeah.
They're distributed randomly along the length of the DNA.
Okay.
All they can ever do is push down to the bottom and stall each other, and you're going to have to get it. So it turns out any reasonable model will give you a Boltzmann distribution, roughly speaking, exponential distribution of loops. To get these regularly sized loops, you need them to dissociate. I'll show you how this works in a second. So what's great about this potentially achieves this kind of local compaction without cross-linking, because these guys, remember, they're bound to just this DNA molecule. They only have an opportunity to bind here when it's the same DNA molecule. And these loops are in good solvent conditions, and so two of these things brought together are two polymer brushes, which will tend to push away from each other. And it turns out that there is some other type of data I'm not going to talk about today from biology, which is actually quite consistent with this kind of loop extrusion picture applied to another SMC complex called cohesin. Well, the bad side of this was entirely science fiction, this loop extruding function in 2016, but the situation has changed dramatically in the last 3 years. All right, so this is just an idea, Dave, of what happens to the loop size if you have motors binding and then extruding loops without ever dissociation. This is how the loop size distribution looks as a function of time. You get kind of a lot of small loops that fluctuate in size. But if you allow them to dissociate, 3 different simulations, if you allow them to dissociate and rebind, what happens is one loop sort of takes over and the loop size becomes large and robust. All right, so, and I've already showed you. All right, and I'm not going to go into a lot of detail about this. For lack of time, I want to get to the sort of discussion of the nucleus as well. So, in fact, we teamed up with Leonid Murney's group at MIT, and they're very good at doing very large-scale simulations. And this is exactly this process applied to a polymer, which starts out as a kind of a compact, highly sort of entangled and compacted coil. And now what's happening is SMC complexes, shown in red, are binding and translocating and then unbinding. And that's all they're doing. And the polymers can pass through each other, so it's as if we have topo 2 present. And you see what happens. You have breaks along the length of this protein scaffold. There is no protein scaffold in the usual sense because the proteins don't stick to each other. Nevertheless, this thing self-organizes regularly sized loops. This is the loop contacts along this polymer, the multiplicity of SMC complexes at the basis of those loops. And even at this rather low level, you get a nice noodle organization, which is obviously a lot stiffer than the initial polymer was. There you can see the persistence length is sort of at the scale of these small bends along the polymer. The emergent structure is a bunch of SMC complexes extruding loops, competing with each other and pushing on each other at the bottom of the loop, but because they can dissociate, you can have formation of this nice regular loop structure, and after as time goes on, the polymer actually pushes away from itself and becomes a kind of a superpolymer with a much longer persistence length than the original polymer. So to give another idea of how this looks, all right, there's no condensation of any kind here. Now we're just looking at the 2 polymers. Red and blue represent 2 polymers, which are actually attached together at one point, much like the 2 copies of a chromosome are. The 2 chromatids are attached together very firmly at the centromeric region of the chromosome in the center. And what will happen is, again, in the individual polymers, the red and blue polymers, are being organized and folded along their length by this lengthwise compaction process driven by loop extrusion by condensins. And the result is 2 large-scale polymers, which are now folded. You don't see the SMCs and the condensins in this simulation, which are folded into thick, self and mutually avoiding structures, which actually sort of push away from each other. So this is a mechanism, we think, by which chromosomes are actually segregated from one another. All right. So this was still sort of science fiction until a series of very nice experiments, the last of which, most recent I'm showing you here, from Case Decker's lab, where actually it's now been observed that condensin from yeast is able to, this is a piece of DNA, which, if we run the movie again, that starts out being a piece of DNA being stretched by flow. A condensin binds here, and it's able to tense up the DNA with forces of up to about half a piconewton by apparently extruding a loop from the location of where it bound. Okay, so this has now been really observed to take place, and these loops have been observed to be apparently extruded by yeast condensin. This is a process which is obviously ATP dependent. This is not going to happen naturally. They're going to tense up a polymer. You see tension building up in a polymer by some thermal kind of effect. So this is the ATP dependent process. How do we think this actually works? Well, we imagine this works by coupling the shape of the enzyme, which we know goes through a kind of a 3-state cycle. With no ATP bound, we know from bacterial SMCs that there's an initial state where the 2 arms of the SMC are actually sort of closely associated, and there's a chamber down here. Then when ATP binds, we know that this chamber, there's an upper chamber that binds, and the ATPs actually bridge this boundary between the upper chamber and the lower chamber. And then when ATP is hydrolyzed, ATP molecules are broken, this chamber opens up, and then the thing goes back to the non-ATP state. So if we couple, we also know from other experiments that the DNA probably goes through this lower chamber. If we then couple, look at this arrow on the DNA, loop capture, which we know takes place by these enzymes, presumably involving DNA binding sites up here at the top, through this chamber, we can then, when ATP is hydrolyzed, release this lower piece of DNA, and then when this thing folds back up, rebind somewhere along the length of the DNA over here to the original DNA. And this type of model actually, because of this loop capture, has a very low stall force, very similar to what's seen in the Case-Decker experiments. This is kind of like what we imagine the translocation and loop extrusion functions seen by Case-Decker's lab, how it works. All right, and I was very happy to see people at New York University are very enlightened, and I was happy to talk to Andres today, and he's recently written a review article on condensin using the approved language of compaction, not condensation. There's no surface tension acting here. There is compaction of DNA actively by these molecular motors. At least that's our current picture. All right, very good. So I wanted to talk for a few minutes about nuclei. So I have a very talented postdoc named Andrew Stevens, who unfortunately now this rather likely has to be changed to 100% likely he's going to a faculty position. He's done fantastic work in my lab isolating nuclei from mammalian cells. This is all work in either mouse or human cells, a lot of work in human cells. And Andrew came to me from yeast genetics lab and wanted to work on mechanics, wanted to learn about biomechanics, and also wanted to do experiments on individual nuclei. So experiments before Andrew's had largely been done using sucking of nuclei or of the cell, including the nucleus, into glass pipettes and using the nucleus as a kind of soft piston to do mechanics measurement. So this works, and you tend to get dynamic information. The compliance of the cell nucleus is a function of time as the nucleus gets sucked into the pipette. And you know, these experiments are indicative and helpful, but they're not giving us the kind of physics type information we'd really like of reasonably uniform strain and stress across the nucleus, because of course the region that's being highly stressed, highly strained and highly stressed, is the region that's being pulled into the pipette. So Drew's idea was to, okay, let's use the pipette system to do more physicsy, more uniform strain experiments on the nucleus. And so, okay, so he set up experiments like this. These can be done to some degree inside living cells, but most of our experiments are done for nuclei extracted from cells. We've done enough experiments in living cells that we think the results are applicable. So, okay, we're moving one pipette and watching the other, the pulling pipette moves, and we watch the force pipettes move, bend, and then we can calibrate that bending. Okay, good. So what do we see in those experiments? I'll just briefly bring your attention down here. First of all, this is a whole nucleus with human nucleus with 46 chromosomes in it, and it's about 10 microns in length before we start stretching the nucleus. And this is the change in length down here, and this is the force in an experiment. What we see are two regimes. There's an initial regime where the force goes up with extension, and that is to about 30% strain. We've increased the length of the nucleus by about 3 microns or 30%. And then apparently you cross over to a secondary regime. And then there's very, very slow hysteresis, but if we sit at the bottom at the end of this experiment for a few minutes, we can go back up to along the same curve. Okay? And so there's a distinct initial spring constant and from a secondary spring constant. So being physicists, we would say, well, maybe the origin of this spring constant is a different spring from this secondary spring constant. Okay? And so nucleus is a complicated object, and one can immediately ask, what happens when you take the chromatin component away? All right, good. So we can do that with enzymes. So for example, we can use an enzyme called micropipettes nuclease. It cuts basically quite indiscriminately, will cut up the interior chromatin, the DNA inside the nucleus, into tiny bits. And actually those tiny bits tend to escape the nucleus. We actually lose about 60% of the chromatin from the interior of the nucleus in that type of experiment. And what you're left with is a nucleus that started like a kind of a round ball, and then at the end, it's become, actually round ball is like up here, and then it's become a kind of ghostly thing that when we stretch it out, you can almost not see it at all. Okay? So here's a result of that kind of experiment. The initial pre-enzyme treatment spring constant is like this, at initial extension 0, it starts to come up with this kind of like nanonewton per 3 micron spring constant. But then after micrococcal nuclease treatment, it's gone completely, the initial behavior. But despite this ghostly look to this thing is still very strong. The nuclear envelope still has the spring constant similar to the initial value of the nuclear envelope spring constant. So what this tells us is that, first of all, the nuclear envelope is quite strong. This is really the first time that I think people have directly measured the mechanical stiffness of the nuclear envelope. But also, up to this 30% point where the nuclear envelope starts to become stretched, you have essentially the chromatin in the nucleus is the thing that's bearing the load. So it's the DNA that's actually not just the genetic component of the nucleus, but is the mechanical component of the nucleus. All right, so this is what this lamin network looks like. It's a network of proteins called intermediate filaments, which are in the nuclear envelope. This is the thing that's bearing the load after we take the chromatin away for more than 30% stretching of the nucleus. And so it's a kind of a network, cross-linked network of polymers that's embedded in the cell membrane that forms the boundary of the nucleus. And so that thing is a solid polymer 2D network on the surface, you can imagine, of a sphere. And then in the interior, there's a 3D polymer network, the chromatin. So it turns out Ed Bannigan and my group did nice simulations showing that indeed, when you make sure that you cross-link the interior chromatin together and stick it to the interior of the nuclear envelope, both things we know are going on inside the cell, then we get this two-stage elasticity, where initially the chromatin is bearing a load, and then secondarily the nuclear envelope is bearing the load. Here we cross-link the exterior. Balls are green, they represent the lamins, and the interior chromatin, the DNA and nucleosomes and histone proteins and other stuff are linked together in the interior. If we strengthen, leave the shell the same, but weaken the polymer, we can kill that initial spring constant. If we take it all away completely, we retain the secondary spring constant, like in the micrococcal nuclease experiment, and it behaves basically very much like the experiments. All right, so that works pretty well. But now, what you guys might be thinking is that, we can change the character of the chromatin in the interior of the nucleus quite strongly. You guys have probably heard of epigenetics, and the type of epigenetics that I'm going to be talking about here is modification, chemical modification of the nucleosome proteins so that they stick together more or less. Proteins that stick together more, the nucleosomes that stick together more, it's mediated by other proteins inside the nucleus, are called heterochromatin, and they organize into a shell around the outside of the nucleus, and also you can fill up the interior of the nucleus. This red stuff is representing heterochromatin in a particular experimental work by one of our colleagues at Northwestern. And we have drugs that we can use to drive the chromatin in the nucleus towards being more acetylated, or euchromatin-like, and soft and decompacted, or towards heterochromatin, which is, you can think of like cross-linked rubber, okay? More methylated, more compact, stiffer, okay? And so what we can do is we can use drugs, and so this VPA thing is a drug called valproic acid, which promotes buildup of acetylation and removal of methylation. Turns out the same amino acid location is used for the types of methylation and acetylation that are relevant to the mechanics of the nucleus. So when you add acetylation, you automatically take methylation away. So in fact, VPA is a so-called histone deacetylase inhibitor, and it inhibits deacetylation of histones. So you tend to build up a lot of acetylation of histones. And indeed, we can see on average, our spring constant goes down after treatment by VPA and TSA. There are other drugs that will increase the amount of methylation. Something called methylstat is a histone demethylase inhibitor. It tends to build up methyl groups on nucleosomes. That brings up the spring constant. And you can see there's a lot of heterogeneity in these types of experiments, so Andrew had to do a lot of experiments to see this. We have done this in a variety of contexts. All right, so we can change the stiffness of the nucleus using drugs that change the chemical character, can change the specific chemical marks on the nucleosomes in a rational way. There are a lot of diseases that are associated with funny shaped nuclei. Okay? A normal nucleus is in a normally growing cell is usually fairly round. I'm sure Alexandra has shown you lots of pictures of nuclei. But there are a lot of disease states where you have very poorly shaped nuclei, including a lot of cancers. In fact, the Pap smear is basically a diagnostic which is looking for irregularly shaped and textured nuclei. So there's a lot of nuclear shape dysfunction associated with disease. And there's a lot of forces acting in the nucleus. There's the nuclear envelope itself confining chromatin in the interior. There are microtubules pushing on the nucleus, and there's the actin cytoskeleton, which is tensing the nucleus by being polymerized around the nucleus. So we might ask, can messing around with these histone marks epigenetically control the geometry and stability of the nucleus? And the answer to that is yes. We can, by drug treating nuclei, change them from being nice and round to actually undergoing what look like mini explosions due to elastic mechanical failure of the boundary of the nucleus. This can be quantified. So here's a nucleus. This pink stuff is the chromatin. The green stuff is a protein which normally will stay inside the nucleus, but this nucleus is going to burst at this bleb point, and you'll see a release of green into the surrounding cytoplasm as the contents of the nucleus is released into the mobile contents are released into the cytoplasm. This is very bad, exposing nuclear contents to the cytoplasm and cytoplasmic contents coming into the nucleus, and causes a lot of DNA damage. And so what we can see is that if we treat normal cells with drugs like VPA and TSA or yet another epigenetic drug, we can increase the percentage of blebbing in nuclei, just count up the number of blebs in nuclei, dramatically up to levels similar for actually making the lamins themselves defective. And these blebs tend to be along the major axis in the most curved region of the nuclear envelope. And there are a number of people that are working on models really based on elastic stiffness in the nuclear envelope, which is controlled by the shape of the nucleus. And so we see a strong enhancement of total blebs along the long axis. We can take patient cells from this disease I mentioned very briefly, progeria, which have defective shaped nuclei, and this is obviously an anecdotal picture. After treating them with epigenetic drugs that build up heterochromatin, we can round up the nucleus, and so that can be shown the irregularity of the nucleus. The percentage of bleb nuclei goes down dramatically after treatment of the patient cells by methylstat. So we're not saying we can cure progeria using epigenetics, but we can change the phenotype of the disturbed nuclei with these types of epigenetic drugs. So what the bottom line is for the nucleus, we can take a normal nucleus, it can become weak and prone to blebbing and rupture by increasing the amount of euchromatin, of soft euchromatin, or driving down the amount of strong heterochromatin. And we can take cells that have problems with their lamins and rescue them by cranking up the amount of heterochromatin. Funny shaped nuclei we can crank up as well. And we now understand, I'm not going to have time to talk about it today, but we can now control this extracellularly. We now know that the cell can sense through the exterior plasma membrane, in particular can sense stress, and when the cell membrane is under mechanical or osmotic stress, we now have carved out a signaling pathway which leads to, among other things, changes in mechanics of the nucleus in the logical way. So if you beat on a cell with a hammer, it will increase the amount of heterochromatin in the nucleus, apparently, probably as a stress protection kind of mechanism. So Andrew will be working on that at his new job. All right, good. Now I won't talk about epigenetics and metaphase chromosomes. I'll just finish by saying that, well, what I've told you a little bit about is the human nucleus. It's a two-component elastic object, a gel, 3D gel in the interior, the chromatin, in a flexible lamin bag, and has strong mechanical hysteresis. We can tune the stiffness epigenetically, and shape and rupture of the nucleus can be controlled epigenetically as well, and we think even extracellularly. And I started out by telling you that metaphase chromosomes are lengthwise compacted. We think this crosslinker is condensin and operates by loop extrusion to accomplish this topological and geometrical folding of the metaphase chromosome. Thank you very much.
Questions, please?
What is PTMs at the end?
Oh, you know, so biology is a land of TLAs. You know what a TLA is?
No, I know LSF.
It's a three-letter abbreviation, right? So, in fact, the situation is so bad in biology that now you start to run out of phase space for TLAs. And so, for example, we study a protein called BAF that's associated with the nuclear envelope, BAF1, and there's another BAF that's completely unrelated. And this has happened several times to me. So even you have to go to some higher literacy. Okay, so PTM, post-translational modification. So you know, the genes in your genome are not the whole story. The genes define the amino acid sequence of the proteins that you can make. But then there are legions of other enzymes which are dedicated to changing those amino acids chemically by covalently sticking things like acetyl groups and methyl groups and phosphates and other proteins like sumo onto the amino acids. So those post-translational modifications are called post-translational modifications. They follow protein translation and folding. So proteins can be in many, many different sort of chemical and information carrying states.
But jokes aside, I don't understand what you're saying here. I understood that you insist that this compaction happens because of
Yes.
loop-extrusion and so on, but then somehow you relate this to PTMs.
Yeah, okay, well that's the slides I didn't talk about.
Oh, I see.
So there are undoubtedly other things going on in parallel other than condensins. So the simple story is good. You're a good student, you learn the simple story that condensins are folding up chromatin in a lengthwise fashion. But many of us suspect that there are other things acting in parallel. So when you buy a car at the car store, you can take it home, but you can have an option to have a turbocharger put on it, right? And it'll work without the turbocharger, but you could win more races if you had the turbocharger installed, so you get the turbocharger. So there are probably, we think there is a role for nucleosome interaction to play a role in stiffening up the metaphase chromosomes. And we don't really understand how this works, but we have done experiments. We recently published a paper showing that the stiffness of metaphase chromosomes, we can double the doubling force, double the Young's modulus, by heavily methylating the histones. So somehow those methyl groups can contribute to overall chromosome stiffness. And there are a number of aficionados that are working on this kind of problem, understanding how histone post-translational modifications may be impacting metaphase chromosome organization. But we can clearly see this. Interestingly, this experiment was motivated by a desire to see what happens with acetylation. So here's the acetylation going up, 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 but surprisingly, there's no change in the mechanics. So acetylation, making apparently the chromatin less compact, it's actually showing that the good solvent property is very much there. What's probably going on in the methylation case is that HP1 alpha is then binding on the metaphase chromosome and acting as an additional crosslinker. That's what we suspect. It's a very poor crosslinker. We see it dynamically leaking off metaphase chromosomes in those experiments. Good.
Thank you.
Yes,
Yeah. In what extent a chromosome can be reconstructed from a component?
So, as you know, as I'm sure you know, Alex, people can assemble things that look pretty much like metaphase chromosomes using mixtures of proteins that are extracted from things like xenopus eggs, okay, oocytes, right? So there you have a large number of proteins. You know, condensin and topo2 are very important in that process. You can chemically mess with them and take them out, and you will not have the proper folding of DNA into metaphase-like chromosomes. But do we know exactly which proteins are the required set for that to take place? Well, I would say we probably don't understand yet. So Tatsuya Hirano in Japan has been working very hard on this problem. Maybe you've been following this literature. So he has been making progress towards folding chromosomes. So the way this experiment works, the dream experiment is you go to the chromosome protein folding store and you buy purified protein A, maybe condensin, right? And purified protein B, maybe topo2, and then some set, minimal set of proteins, maybe 2 A through G of highly purified protein. Then you take a piece of chromosomal DNA. Actually, in these experiments, you usually use the sperm, which is low in protein content from the same species, or maybe a sperm from human DNA isolated from human sperm. People have cross-reacted these kinds of things. And you mix everything together, right? And add ATP. Don't forget to add ATP. Right buffer, stir, and you get a nice folded chromosome. Okay, we're not there yet. But Tatsuya Harano has been making steady progress, and it's very clear condensin is a required component for these types of reactions. It's very clear topo2 is a required component for these reactions. Beyond that, I think it's poorly understood exactly what state histones are required. I mean, people can see perturbation of metaphase chromosome structure with different types of histones, but you know, I think it's still very poorly understood exactly what the minimal set is to really get to those nice-looking metaphase chromosomes that you can get with the full extracts. The purified protein experiments tend to produce things which are somewhat compacted, but they haven't gone all the way towards the sort of metaphase-like compaction. It's an interesting condensed matter physics problem, just taking a polymer and compacting it along its length using locally acting enzymes so as to get a well-regulated thickness to this rope, which is made of, you know, a long string of DNA, we know, linearly arranged from one end of the chromosome to the other. So the loop extrusion hypothesis is one solution to that problem. Obviously, I hope it is the correct solution, but hope only gets you so far in this business. So we'll see if there really is an explanation of that.
We have one more question here. So.
I missed the point why the
Yeah.
concatenation number is inversely proportional to the number of chains. Often I would expect the opposite.
So it's simply because my definition of that catenation number is the catenation between any pair of chains. And so the near collisions between yourself and another chain, if you have little n chains, well, you know, where the chains are long enough so you can use this kind of mean field polymer argument, each crossing will be with 1 over n of the other chains. That's all. It's just definition of the catenation number. It's the catenation number between any pair of chains. The total catenation number of you with everybody else will be n, right? As you're probably thinking. That would be the total entanglement you would have with other polymers. But still, yeast appears to budding yeast appears to beat the entanglement problem, at least in part by having a lot of short chromosomes.
Okay, last question, Andreas, and then we'll continue with wine and cheese.
Okay,
I was just
the
wondering,
visionary, Andreas Hofwagen. Okay, good.
I was just wondering, when you pulled out the chromosomes
Yeah.
at the beginning, you showed these very thin fibers
Yeah.
that you observed. What are those? Is these transformers not working?
So we see these, they're a real annoyance because like every biology book, you know, you study elementary biology, you know, my course in high school, we learned that, you know, the chromosomes are separate from each other at metaphase, right? So, okay, these are slightly pro-metaphase, so they're not quite all the way to metaphase because if they're attached to the spindle, they're hard to isolate. They're still pro-metaphase chromosomes. So, there's actually an old literature of studying these things, and there's been a lot of, I'd say, controversy back and forth about what they are. But they appear, at least in some case, to be, guess what?
Tangles?
They're tangles between different chromosomes, but they involve the nucleolar genes. So Jen Gerten at Stowers has done some characterization that at least some of these tangles are from the 5 acrocentric in humans, 5 acrocentric chromosomes, and they're the nucleolar genes, just like in yeast. So these could be just very late separating regions of chromatin. As you know, they're repeats, so maybe it doesn't matter if you break them. You know, they get cleaved at the end of mitosis and then repaired. So, and you know, there's significant variation in like cells with hundreds of nucleolar genes, hundreds of ribosomal genes, excuse me, in the actual number of those genes. And there's quite a large variation with time through cell division. So it could be that those are just broken. However, we don't see entanglements between just 5 of the chromosomes. They're all entangled together. So there are some experiments that suggest those are other repeats. Some near centromeric repeats have been implicated in some experiments, but it's still quite open exactly what they are. Yeah.
May I just ask,
Yeah.
hasn't Daniel Gerlich shown, right, that you have the KIFs on your chromosomes? Wouldn't you need the transcription of those NORs to actually have the coverage during chromadosis?
Oh, you mean, I think there's So all of that stuff is made before mitosis, right? All of those proteins, right?
And for some reason, maybe they still need access during.
Yeah, I suppose it's possible. So we're studying Ki67 cells right now. That's what you're thinking of, right?
Yeah.
The Ki67 protein. So those actually are able to it turns out, okay, so again, another supercharger, another turbocharger for mitosis. It turns out you have chromosomes and you have maybe PTMs playing a role, and it turns out that, yeah, any soft matter person would say, hey, what about adding some surfactant, right? Let's add something that sticks to chromatin and likes the nucleoplasm or cytoplasm, and then that should push the chromosomes together apart even more. And yeah, sure enough, there's a protein that does that. It's called Ki67. It's like a diblock copolymer that tends to form a surface layer on chromosomes. So it's kind of interesting. In the absence of Ki67, the chromosomes end up closer to each other and are more entangled with one another, but they are still individualized as individual chromosomes. So you still have the prophase mechanism is going through more or less normally. It's just that latter stage of separation. So in fact, we're looking at the Ki67 knockdown cells right now, and we can sort of separate chromosomes from one another.
It sounds very exciting.
Yeah, it's interesting.
We'll continue with wine and cheese. Please, everyone, to 8th floor. And let's thank everyone. Thank you.
My pleasure.
Cool. So you, I don't know.